Hello guys, we are going to go through lipids, AQA, A-level biology. Okay, so let's begin. So this is the specification, you should know that, and you should be referring to it at all times. So if you don't have it printed off right now, go and print it off, but be sensible with your the amount you print. Do it like an A5 book or whatever, but you need to have it in front of you. Reason being is so that you can tick it off as you go along. I will be going for the whole specification, but it's for your own sake as well, and you can annotate stuff, and you can be like, oh, I don't know what this means, oh, I know what that means, tick, blah blah blah. Annotate it, it's yours, and it's obviously for your benefit, and examiners are only going to be testing you stuff on the specification. So let's begin. Lipids are different to carbohydrates because they are not polymers. Now, carbohydrates, quite a lot of these are polymers. Now we've got starch, just as an example. We've got amylose pectin, we've got cellulose, we've got glycogen. So there's quite a lot of carbohydrates which are polymers, but lipids are not polymers. They are not polymers. That's the main thing that you have to engrave into your head. They are not polymers. And the term lipid covers a group of substances that include fats and oils, which are types of underneath the category of triglycerides. And then you've got things like steroids, which are also examples of lipids, and you've got sterols and waxes as well. Now, triglycerides and phospholipids are the two main types of lipids you need to know about. Don't worry, I'll cover these in a lot more detail. So what are the roles of lipids? Just like generic lipids, what are the roles? So let's have a look. So number one is the source of energy. So both triglycerides and phospholipids can act as a source of energy where they are used in respiration. How? I'll cover that in year 2. You don't need to know it anyway, but for example, just for the sake of it, I'll go for it, but in the year 2 videos. Then we've got waterproofing as well, so it's in the name, waterproof. Doesn't like water, doesn't interact with water. Well, kind of, but just generally doesn't interact with water. Third, insulation, flexibility for cell surface membranes. So let's have a look at this. So triglycerides are formed by the condensation. You should know that word by now. If you don't, go back to my first video or second and learn the definition. So triglycerides are formed by the condensation of one molecule of glycerol and 3 molecules of fatty acid. So what on earth does glycerol look like? Bear in mind, right now we are looking at triglycerides specifically, so you can forget about phospholipids for now. We are looking at triglycerides. So what does glycerol look like? So just a little key hint, every time you see the suffix ol, always think alcohol. So this is a type of alcohol, and this is what it looks like. So you've got 3 carbons, then you've got 3 hydroxyl groups, and then the rest are just hydrogens. Bear in mind, carbon forms only 4 bonds. And yes, you do need to know the structure of this glycerol. So you can see carbons formed 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, and 4. So yeah, you need to memorize that. And what does a fatty acid look like? So it looks something like this. So you've got so this is one fatty acid molecule. So you've got a carbon with a double bond to oxygen, and then a single bond to this hydroxyl group we have here, and then this thing called an R group. Now you're probably wondering, what the heck is an R group? Well, an R group is anything. So basically chemists, biochemists, all these scientists people, they want to abbreviate long, massive, complex equations. I say equations, I mean like formulas. So what they do, they just substitute R, because the main bit which makes up a fatty acid is this portion here. And then you can add anything else you want in this R group, and it will still be a fatty acid because it's got this very important bit which makes up the fatty acid. So I can have this as a fatty acid, so I can have another carbon, so I can have this is also a fatty acid because it's got this certain group here which makes it a fatty acid, and this bit here is referred to as the R group because it's not this. So hopefully that does make sense. So here is a better diagram. Now ignore this bit for now, just focus on this. So you've got your glycerol, 3 carbons, 3 hydroxyl groups, or you can say alcohol groups, and then you've got your 1, 2, 3, 4, 5 hydrogens attached to the carbon. Then your fatty acids, so you've got your carbon, which is double bonded to an oxygen, which is then got a bond to a hydroxyl group, and same again, so you've got this another fatty acid, and you've also got this R group as well, which can be anything. So now we've seen the ingredients we need to make our triglyceride. So how do we make it? Okay, we've got the ingredients, so how do we make it? So you need 1 glycerol, you need 3 fatty acids, and then you add them together, and voila, you've got your triglycerides. So you've also got to know how to draw this triglyceride right here as well. Let's actually take a look how we can form this together. So I've drew part of a glycerol molecule right here, and I've drew the whole fatty acid molecule, and I've got the R group right here as well. So what happens is we draw it out like this, and obviously to make a triglyceride we have to go through this process called condensation. So in condensation we have to release a water molecule. So this is so we get our water molecule, which is this H2O. This is released, and then as a result this oxygen bonds to this carbon here, which makes this section right here. And then obviously you do that for the second carbon, and then the third, and then hopefully you should get a diagram which looks like this. So I'd have a go, experiment, do it over and over again until you get this right, because it's really crucial you do get this right, because they could ask you in the exam to circle the fatty acid portion of a triglyceride, and you're gonna be like, oh, I don't know, but you've got to know. So, or it could ask circle the glycerol portion of the triglyceride, so you could circle this bit here, etc., and so on. They can ask quite a few stuff, so it's good that you should revise this and go over it and draw it out and memorize it. So I mentioned before how we've got a fatty acid right here, and we've got this thing called the R group. Now I said the R group can be literally anything. It's like an easier way of symbolizing a massive long chemical formula. So we could have carbon, 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 carbon. Okay, you get the idea. So we can have this massive long carbon chain and attached to the fatty acid, and realistically no one's going to be bothered to draw all of this out, so we just represent it with the R group. Now we've got 2 different types of R groups. One is called unsaturated fatty acids. Okay, sorry, I meant we've got 2 different types of fatty acids. One is unsaturated and one is saturated. Now obviously, because this can be anything, and this is the only variable that changes in different fatty acids, therefore this term saturated and unsaturated is referring to the R group. So let's actually have a look what these two are. So unsaturated fatty acids are R groups or fatty acids which have an R group, and within that R group they have a carbon-carbon double bond. And saturated fatty acids are fatty acids which has an R group which only have carbon-carbon single bonds, so they have no double bonds in saturated fatty acids. And the way I remember it is, well, saturated is a shorter word than unsaturated, and therefore it will have less bonds. It's weird ways of memorizing it. Find out your own way, or use my really daft way. Like it probably doesn't even make sense when I say it, but hopefully this idea of unsaturated fatty acids and saturated fatty acids makes sense. So let's have a look at the different properties of triglycerides for now. Let's forget about phospholipids because we're not concentrating on that for right now. So what are the different properties of triglycerides? So triglycerides are mainly used as energy storage molecules, so they contain a lot of energy. So as we saw before, a simplified version of a triglyceride is this. So you've got your glycerol molecule, and then you've got your free fatty acid chains, and each of these bonds contain energy. Every time you break it, it has a lot of energy. And one thing I kind of forgot to mention was these bonds that we form right here between the fatty acid and the glycerol. These bonds have a specific name which you have to know as well, and they are called ester bonds. So another feature is that they are insoluble. So I mentioned before there are examples of triglycerides which are like fats and oils, and as you know, if you mix oil and water, do they mix? No, they don't. So this is another feature. They have a hydrophobic region only, so kind of linked back to this. So hydro means water, and phobic, well, derives from phobia or linked to phobia, fear, scared. So so this triglyceride doesn't like water, it hates water. So this whole thing is hydrophobic, it hates water, hence why it is insoluble in water. They bundle together as insoluble droplets when in water. They have free ester bonds, so let's have a look where those ester bonds are. So we've got our glycerol and we've got our free fatty acids, and the point at which the fatty acid joins onto the glycerol, that is our ester bond. So we've got 1, 2, 3. And lastly, they contain carbon, hydrogen and oxygen only. So I say only, that's true, it does only contain these three elements only, but in other words, what I'm trying to get at is, don't say that they contain phosphate, and I'll tell you why later on. They do not contain phosphate whatsoever. Doesn't have anything to do with triglycerides, and that the reason why I say that is because phospholipids, you can probably guess, they contain phosphate, so I don't want you to get them mixed up. All you've got to know is that triglycerides contain only these three elements: carbon, hydrogen and oxygen. But let's look at phospholipids. So we've done triglycerides, we can put that to a side now. So in phospholipids, one of the fatty acids of a triglyceride is substituted. So we know a triglyceride looks like this. This is a very simplified version. So it's got three fatty acid chains. Here on the specification, it's saying that in a phospholipid, one of the fatty acid chains are substituted by a phosphate containing group. So it will turn into something like this. So it looks something like this. So this is a phospholipid. One of the fatty acid chains have been substituted for a phosphate group. hence leaving 2 fatty acid chains, 1 glycerol and 1 phosphate on this phospholipid, making a phospholipid. A bit of a mouthful to say. So when in contact with phospholipids, when the water is in contact with the phospholipids, they arrange themselves in a double layer with the hydrophobic tails pointing inwards, so they don't like water, that's why they point inwards to go away from the water, and the glycerol phosphate hydrophilic heads pointing outwards, so the glycerol phosphate head is pointing outwards. And hydrophilic, kind of a new term, basically means water liking, it likes water. It's because of the phosphate head that the phospholipids develop the feature of being hydrophilic. Having both hydrophobic and hydrophilic features is referred to as amphipathic. And phosphate has, hopefully I'm right, has this charge here, and water is also charged, and charge-like substances interact with each other, and that's why they mix with water, and that's why they develop this hydrophilic feature. So the different properties of triglycerides and phospholipids. So we've done triglycerides, let's focus on phospholipids. So the different properties of phospholipids. So number 1, it forms cell membranes, allowing movement of non-polar small molecules. Phosphate heads are hydrophilic, therefore attract to water. We've already mentioned that. Fatty acid tails are non-polar, so they don't have a charge, therefore repel from water. It contains a glycerol, contains carbon, hydrogen, oxygen, and a phosphate, and it has an ability to form a bilayer or a monolayer in water. Now we've got a test for lipids, so we had a test for carbohydrates. Now we're going to look at a test, we had a test for starch as well, so now we're going to look at a test for lipids. So if your assumed lipid compound is a solid, then you first must crush it up. So they might have seeds which contain lipids. You must crush up these lipids, seeds, or seeds that contain lipids, and then put this substance in a test tube. If it so put it into a test tube, and then you're going to add 3 to 4 centimeter cubed of ethanol, then add an equivalent amount of water. So ethanol goes first, water goes second, and then shake the test tube with the bung on. Obviously you don't want it to go everywhere, and observe for a white emulsion for a positive test for a lipid. And the reason why we must add ethanol first is because obviously all organic compounds are soluble in ethanol and not necessarily water, so we have to like break some of the bonds and stuff and like get it to mix. So hopefully that made sense. If it didn't, comment down below. If it did, comment down below. And next bit we will look at proteins.